So good afternoon, everyone. I'm Andrew Siebel, the platform manager of MDAP. Today, the team and I will be presenting a few slides for you on our upcoming call for expressions of interest for our MDAP collaborations. I will share my slides with you. And presuming you can see that okay, Ma? I'll let you take the floor.
Not yet.
You can't see it?
Nothing shared yet.
No, I can't see it.
There it came.
That better?
Yep.
Yeah, excellent. Okay. So I'm going to throw to you, Ma, if you would like to take it from here.
Cool. So we're the Melbourne Data Analytics Platform, and here we're going to just take you through our operating model and how we work, and give you some insight into how you can work with us if that's something that you're inclined to do. And then we'll present some case studies of examples of successful projects that we've already been involved in to give you kind of an idea of what it is that we can do. Next slide, please, Andrew. So MDAP is part of the Petascale Campus Initiative, or PCI for short. And the PCI was formed a few years ago to support an academic-led vision of data-intensive research at the university. So it's a 5-year plan to accelerate the university's capacity for this research. And it really has 3 pillars. So I'll start with the leadership component. It's academic-led, and so it's taking academics' voices up to the highest levels of decision-making within the university. So it's a conduit to communicate to the main strategy people within the university and government people what it is that researchers and academics need from the university. So in that sense, they also make a lot of the decisions about what infrastructure is going to be purchased within the next 2 to 3 years. So also bringing the voice of academics into these strategic choices and purchases to decide whether we're better off buying more high performance computing or cloud computing, or where the money is best spent to satisfy the needs of the researchers in the university. And the third pillar is the workforce component, which is us, the Melbourne Data Analytics Platform. So it's formed as part of the PCI, but there's a slight difference. So the PCI is completely funded by Chancellery, and MDAP, or the Melbourne Data Analytics Platform, is co-funded by Chancellery 50%, and all of the 10 University of Melbourne faculties, the other 50%. So we aim to work broadly across all of the faculties within the university. Next slide, please, Andrew. So this beehive figure was provided by Rachel Ramsdale from Chancellery. And it provides an overview of the 23 platforms that exist across the University of Melbourne research infrastructure landscape, most of which are supported primarily by MCRIP, or the Melbourne Collaborative Research Infrastructure Program. So many of these platforms are equipment or facility-based platforms with a service-based approach. MDAP is a little bit different, and there are others here that are also more like MDAP. So it's a people-based platform with a collaborative approach. So we don't offer any infrastructure or equipment or microscopes or services. What we offer is technical skills, and we want to work with you collaboratively to take your data to the next level. So the uni has made a considerable investment in this area, as you can see here, with a workforce of nearly 170 full-time equivalent. Yeah, MDAP is, I guess the important point to take here is that MDAP is here to collaborate with you on your research projects, but also to help you navigate this very complex digital research landscape and where to go for different kinds of problems or questions that you might have. Yeah, next slide, please, Andrew. So the Melbourne Data Analytics Platform is a team of academic specialists working to enable data-intensive research across the disciplines at the university. And we also have these 3 pillars of engage, or ways we operate, I guess. We're an academic unit of highly skilled researchers in all sorts of digital methods, as well as data management and stewardship. So whereas PCI has kind of a top-down approach of working with the highest levels of strategy, government, and decision-making within the university to benefit researchers, we're on the floor working directly with researchers in a bottom-up approach to to feed that anything that we encounter on the ground back up to these high levels of decision making within the university. So we work directly with researchers and groups. We don't work with faculties or specific departments. We work with any researcher that approaches us. So the second part, computing, disciplines, and Noel is going to talk more about that later. We're all experts in different digital methods or computational methods. So we're all researchers. We all come from different backgrounds of academia, and we're uniquely positioned to understand both the language of researchers and computational science, for example, infrastructure providers. So this is really where we add value. We're not biostatisticians. We're not, we don't duplicate a lot of the efforts that are already going on in the university. We add value by focusing on the computing part of working with data. So parallelizing complex code, optimizing code to run faster, using high performance computing, using cloud computing. All of these things are the things that we really specialize in. And because we're academic specialists, we're employed as academics, we're researchers, our ultimate goal is to achieve research impact, similarly to many of you. In the form of publications, research software, but we also value other non-traditional and qualitative academic inputs as well. So we have a little bit more of a broader definition of value. Next slide, please, Andrew. So this diagram was generated for a recently distributed brochure on MDAP, and if you haven't read it yet, I recommend it. It gives a really nice overview of some of the things that we've been involved with in the last 12 to 18 months. And this diagram in particularly shows the cross-faculty connections and interactions to business services that we've been able to facilitate over the last 12 to 18 months. So on the left, you can see kind of a close-up of medicine, dentistry, and health sciences, FVAS, and the Melbourne School of Engineering. And the different colors represent, so the gray is data science kind of contribution, so building a machine learning model or things like that. The darker green is computation, so things related to high performance computing or cloud computing or things like that, more infrastructure based. And the lighter teal is data stewardship, so that has more to do with ethics and data governance and policy and procedures. So I'll throw over to Noel to tell you a little bit more about the team and what our specific expertise lies in. And I'll be back with you later with some case studies. Thanks.
Thanks, Ma. So I'm Noel Foe, Senior Data Research Specialist. So the team is currently stands at roughly about 20 people. We recently had pretty much a doubling at the start of the year from last year. And the idea is to actually represent the very large church across the university from a point of view of disciplines. So we've got a number of people who come from a very wide range of skills, from archaeology, psycholinguistics, computer science, geography, astrophysics, maths, AI. We cover all the omics in the bioinformatics area. And as a collection across the thing, we've got a depth of and broad range of experiences, both from industry as well as academic. We cover a large range of computational disciplines as well. And like I highlighted before, the academic domain. So we've all got a specialized academic background, but we're very broad across the computational disciplines. And on top of that, across the whole team, we've got a very broad range of organizational skills. Some of the key sort of members, so to speak, within the team, up on the top right of the picture, or top left, is our academic director, Andrew Turpin. And obviously you've met Andrew Siebel as the manager of the platform. And we have Joe, who sits in the fourth position on the second row, who's our comms specialist, and everyone else within that group are data specialists. Next slide, Andrew. So the main role for a data specialist, principally the whole team, is not only to bring our expertise and our background in computational skills across that particular discipline, and help and collaborate with people to uplift what they can do with the data. And we understand that big data means a lot of different things to different people. Some areas, big data is a data set of 20 with a very large number of variables, whereas other people, it could be a couple of variables, but with a data set of over 1 million. We also help and foster research data stewardship, and that's everything from assisting in the collaboration, from collection of the data, some experimental design, how to store, manage, and curate that data, and then, of course, how to interact and work with high-performance computing, and as well as bringing our breadth of skills around data analytics. But on top of that, we also foster collaboration across the university, and will help pull in the necessary skills, such as building a new technique. We would probably not tap on the door of the Centre for Data Science. Next slide, please, Andrew. So like I said, the skill set that sort of comes is very large, and our principal aim is to collaborate with you on both the research design. We can also help you articulate the research question. In some cases, you may have a lot of data and realize that there's a good value in there, but aren't unclear as to how to best articulate the type of data question you want, as well as providing intellectual input into the research. We help write new software, design and other algorithms, and also contribute to the entire data pipeline. So cleaning, imputation, etc. We also are key to introducing people or the collaborations to new technologies, such as the cloud and high performance computing, new techniques and concepts in data visualization, and of course around the data science area, the machine learning, natural language processing, as well as image and video processing, and how to work with software engineers, etc. So it's a very large area, and we're here to really help you get the maximum out of your research objective. Next question, slide, and I think we're handing over to
me. Hello.
Say hey.
Yes, I know. Hi, hi everyone. My name is Zahir, and I am also a research data specialist at MDAP. I'm gonna dive a bit more on the role of research data specialist and what we do every day. So there are 4 pillars into our role that we spend all our time on. We start with the research, the advocacy, and consultancy and community. So as for research, this is what we spend most of our time on. And we do not initiate our own research projects. However, we collaborate on research projects across with collaborators across all the faculties at the university. And I'll talk a bit more about how to engage with us in order to collaborate on research projects. There are 2 ways to engage it. The first one would be on engagement with the major collaboration, a long term between 3 to 6 months. And the other one would be through consultancy. It could be 1 or 2 meetings, or introduce you to a new technology, installing an open source library. So you don't need to apply for the consultancy. You can just do it through the shared inputs, and I'll talk about that a bit more. There are other important part of our role is advocacy and community building. As for advocacy, we collect feedback and the need of researchers, and then we convey that feedback into the PCI, people who are responsible on realizing the digital infrastructure at the university. Community building is an essential part of our role. We exist to break silos at the university, and we seek to build a community of practice of people who have a similar professional roles as us, including data stewards, data curators from scholarly services. Move to the next slide, please, Andrew. So as I said, we spend the majority of our time on research projects, and this is done through 2 ways of engagement. The first one is through formal collaborations, which is done through a formal application process. We open the expression of interest twice a year, and this is mainly for a long-term collaboration between 3 to 6 months. So the unique thing about us at MDAP and this collaboration is that when you apply with to collaborate with us and get selected, you get access to multiple people, a slice of every skills in the team. So you get more than one person to work with you in a collaboration and stay with you between 3 to 6 months, the duration of the project. And usually these collaborations are a merit-based allocation. They are selected based on their high strategic value to the university. And we do not select this collaboration. There is an independent senior academic decision-making committee that would select this collaboration for us. So the application process is very simple. It starts with an expression of interest, which already open, and you can find a link on our website. It's a soft touch online form. So we just ask you very basic questions about your research project. So the EUIs will close on the 15th of November. So we do triage the EUIs and then we get back to you whether you proceed to a full applications. The full application is a couple of pages of Word document. Or we direct you to more suitable department or services at the university. But the majority of EUIs actually get an invitation to submit a full application. So full applications will open in the 26th of November and will close in the 18th of December. Next. So the other way of engaging with us, as I said, is these consultancy projects. It's informal. You don't need to apply. There's no application. You just send us an email to mdap-info@unimelb.edu.eu. And this is meant to be a very short one-off engagement where we have maybe 1 or 2 meetings with you to help you in some open source library or some other digital or technical stuff, or even new research stuff. So it's a short term, and it could potentially, some of these short engagement could turn into an application for collaboration in the next round. Next. So the last round, the previous round 2020A, we received for the formal collaboration, we received about 60 EOIs. There are 7 from MSE, 6 from FVAS, and 15 from MDHS. These EOI were triaged internally by us. 40 of these were invited to submit. Oh, they did actually submit full applications. 10 of the EOIs we directed to an alternative, more suitable service at the university. And we advise as well to 10 other EOIs to merge and submit full application as well. So then the full applications will go into our internal assessment, where we provide recommendation to the research allocation committee. However, the research allocation committee, which consists of independent senior academic, they make the final decision to which collaborations we work on. So out of the 60 EOIs, and then 40 out of the 40 full applications last round, we selected 17 projects, and we are 17 research data specialists employed on the basis of 15.4 FTE. Next. Andrew, please. Yeah. So now we're gonna talk about some examples of successful collaboration from the current round and the previous one as well. I'm gonna start with this project, which I am leading from the MDAP side. It's called the Automating of Image Segmentation and Morphometric Analysis for Accelerating Neuroscience at the Nanoscale. So our collaborators are Dr. Calvin Eber, Professor Janet Keast, and Bergrin Osborne from MDHS. What we're trying to build here is build an image processing computer vision tools for large-scale neuroscience and neuroinformatic analysis that's required by the next generation of neuroscience. The aim of this particular project is to build tools that can identify and measure nerve cell axons in electronic microscope, EM images. And next, please. Here's an example. Yeah, here's an example of an image. So our collaborator has collected lots of images acquired through electronic microscope of tissues from the pelvic nerve that connects to the bladder in rats. This is an example of one image. It's a very high resolution image. This particular image, I think it's about 600 megabyte. It's a very, and they can even acquire a higher resolution, but this is what we worked with. So the important thing for them is to measure. So this image has lots of blobs. These blobs are, what's important for them is 2 or 3 kind of blobs is, as you can see on the left side, it's called the axons. There are 2 types of axons, the unmyelinated axons and the myelinated axons and the myelin. So what's important for our research is to be able to calculate morphological properties of these axons, like their width, their thickness, and all other morphological analysis or properties. However, to be able to calculate that, they need first to locate them in the image. And sometimes these tissues would have tens of thousands of axons in there, both myelinated and unmyelinated. There is no currently process to automatically locate and segment these axons. Therefore, they're doing it manually, and it's a very laborious task. So they literally draw borders around these axons, and then they pass it through automated pipeline to calculate these properties out of these blobs. There are attempts in, recently have been published work in 2019 where they were able to do only myelinated axons and myelin, but not successfully done the unmyelinated axons. So if we move to the next slide, please. So we started with that work from Nature and Scientific Reports 2019. They did develop a network structure. It's based on the convolutional neural network, particularly the UNet, and they were successfully able to segment the myelin and the unmyelinated axons out of the image. However, the model was not generalizable, where it worked on their images, but didn't really work properly on our images. So, but we started with their model, we tweaked it a bit, and then also we improved it to detect the other type of the harder unmyelinated axons. If you move to the other slide, please. And this is our pipeline where this is the ground truth of one of the samples. Our pipeline is able to segment all type of axons, including the unmyelinated, myelinated, and the myelin as well. And at the moment, this is just an early result. The DICE score is between 0.5, 0.8, which is really great, and it can be improved even further. However, this is still work in progress, and of course, we're gonna publish that once we are finished. So this is the output of our pipeline. Next. I'm going to talk about another collaboration from the previous round. It's with Dr. Benjamin Schultz and Adam Vogel, which I also led along with Mar. So this machine learning for clinical and preclinical diagnosis of neurodegenerative disease using speech data. So the Adam Vogel has collected through international collaboration hundreds of audio recording of people with neurodegenerative disease, repeating the phrase pataka, pataka. Apparently through this phase, they hope that they can actually identify the type of the disease, because this disease affects the speech first. Before the first symptoms is the effectiveness of the speech. Sometimes a human cannot really distinguish the symptom. However, through signal processing analysis, where it's done by Dr. Schultz, he was able to extract some features, but stop there. So we came in and we took these features from all this audio recording and we applied some machine learning method on it. And we were able to actually discriminate and also classify the type of the disease that particular patient who recorded the audio might have. So, and Mar is gonna talk more about the method. Yes, Mar?
Yep, next slide, please, Andrew. So this shows on the right-hand side a projection in LDA space for the 3 main diseases that we were concerned, well, two, Friedreich's ataxia and multiple sclerosis, and comparing them to healthy controls. So when we first started working on this data, there were, of course, a lot of methodological issues that we had to decide. And one of the main things that I got a little bit obsessed with is there were multiple speech recordings for each patient. So patients could come in and maybe do 2 or 3 recordings at a time, or maybe they would come in and do recordings over several consecutive weeks or whatever. And so it wasn't a traditional machine learning problem in the sense that the different rows or the different data points that we had were not independent, were not statistically independent. So I started looking a lot into this, and it turns out there's not a very good, well-established approach for dealing with this in cases like this. So we took kind of the naive approach of averaging over the patients, and that already helped us have a much better performance than if we didn't. But then we did engage also with the Melbourne Center for Data Science, and we referred Ben Schultz to Howard Bondell and his team because this seems like it's a good area for growth, for developing methods that can deal with this case where you have multiple data points for each patient, which are not independent between them. Another problem that we had is that there were more clinical features than patients, many more clinical features than patients. So of course we had to do some dimensionality reduction. We started with the standard principal component analysis, but it didn't really do very well. You needed a lot of dimensions in PCA space to be able to actually separate these 3 groups. So then we thought of doing maybe linear discriminant analysis would be better, because this is a supervised method of classifying, which means you learn from the labels first. So you find a 2-dimensional space in this case that best separates the 3 diseases, and then you see how it generalizes to data it hasn't seen yet. And this worked really well, and it helped us get pretty good performance. And really the nicest thing out of this whole collaboration to me is that we ended up writing a book chapter on best practices for machine learning approaches to big data and neuroscience. And we just learned so much from each other and kind of developing this kind of new knowledge and reviewing all the existing methods that are out there and which ones work and which ones don't. And we had a very heavy emphasis on how important it is to start with tidy data in the first place. And this chapter is going to be published in a book next month. So that's super exciting. This was a very enriching and fulfilling collaboration. If we go to the next slide, please, Andrew, we'll talk about another collaboration that's currently ongoing, currently wrapping up, which is with Haroon Verbruggen from the Faculty of Science, but also involved Sebastian Duchesne from MDHS from the Doherty, Tim Entwistle from the Royal Botanic Gardens, and Sui Willis from CSIRO. So those photos at the top are the members of the MDAP team that were working on this. And in particular, I want to point out Bobby, who's the last person there in the photos, who's our bioinformatics expert. So I came to this without having any bioinformatics background, and he led us all to success. So the project was really, what Haroon wants is to advance our knowledge of microbial eukaryotic biodiversity and evolution by extracting organelle genomes, so mitochondria and chloroplasts, from publicly available metagenomic data. If this sounds like a lot of jargon, I'm going to try to unpack it a little bit for you, because this was my reaction to the first time I heard those words. So metagenomic data is when you take a sample, say from a river or from the ocean or from soil, that can have however many different organisms' genetic material in there as is possible, which is a lot. So I don't know, a sample can have, 20,000 different organisms in there. So people do what's called shotgun metagenomic sequencing of this data. So using the standard genomic sequencing approaches, you can still obtain kind of little snippets of genetic material. And then the puzzle is to bring this back together into, you usually can never reconstruct a whole genome, but you can get close. You can use some sophisticated mathematical and algorithmic techniques to see which ones go together and make it a lot bigger than what you can read there. And so Haroon was mostly interested in a publicly available data set of metagenomic data that goes up to about 50 terabytes of data. So if you were to do this process manually of doing quality control, trimming out adapters and poor quality sequences, assembling the genomes, and then being able to link that back to be able to identify which organisms each one comes from. It's just not an option to do it manually. It takes a lot of time. For each one, you're sending a task and a queue to a high performance system. And it would be pretty much 1 FTE of a person for a year. I don't know, maybe. So the collaboration was all about building a pipeline for this workflow. So Andrew, if you go to the next slide, please. This shows a little bit what the pipeline looks like. So we use WDL, which is called workflow description language. So each task or script you write a little script for, and then those get internally submitted to Cromwell execution engine, it's called. This is just the fact that because high performance computing is a shared resource, you can't run things whenever you want like you would in your computer. You have to go into a queue. There are limited resources, and when enough becomes available for your task, then your task goes through. And so the pipeline that we built is we used something called a singularity container, which, if you know Docker, it's kind of similar, but it works on high performance computing, whereas Docker doesn't. And what you can do is you can run this pipeline in a bunch of different computational settings. So it's portable. You can run it in the Google Cloud. You can run it on the University of Melbourne's high performance computing system. You can run it on the Melbourne Research Cloud or AWS or whatever it is. And it's all contained into one thing. It has already all the software that you will need to do it. So you can just set it running and kind of forget about it and then come back when all the results are ready. So the really nice things about this collaboration was that it involved several departments or people from different institutions. And it required really a large breadth of expertise and testing all the different little parts of this pipeline and then putting it all together. And we didn't start from scratch, luckily. Bobby already had a version of this pipeline that he had developed during his work and his previous work at Melbourne Integrative Genomics. So we were able to build on that, add the tools that Haroon Verbruggen was interested in, and make options for what to use for which different type of data. So if you go to the next slide, Andrew, one other output from this collaboration, apart from the pipeline, which is the main one, is we're putting all of this in on a website on an enterprise virtual machine in the university. So anybody will be able to come and see for all of these studies that we downloaded, metadata about each of the data sets, where they came from, be able to track it back and download not only the raw files, but also the assembled files for downstream analysis. For example, in Haroon's case, who's interested in the downstream kind of genomic binning part of it, to be able to learn more about organelle genomes. And so this is still in development. Rob Turnbull from our team has worked heavily on this and has done an amazing job. It's looking amazing. And so I really look forward to be able to share this with you and see if there's anything you can do with the data that's there. And now I'll throw over to Haroon for one last case study. Sorry, to Noel.
No worries, thank you, Ma. So this is our last case study for the session. And this is a collaboration with Professor Robin Gasser, Drs. Pasi Cajon and Dr. Neil Young, who are all on the call. And at the end, after this, I'll get one of them or the team to actually give some commentary. So this is really around taking an existing pipeline that was aimed to find all the variants in a population of parasites, nematode parasites. And these are non-model organisms, so we don't really have a reference genome to actually find these variants. So the idea here was to actually take their existing pipeline and make it run on high-performance computing or other cloud infrastructure, as well as our own sort of machines within the lab in a way that would actually accelerate the research output. If we can move to the next, and that's working with my colleague Bobby, who is also working on the prior collaboration. So the next slide. So this is a very complex pipeline. It was originally written in Perl with a mixture of both R and Bash scripts. It only was really designed for a multi-threaded system and not designed for a highly parallelized HPC environment, which is where in this type of case, you can get a huge increase in speed and performance. So there were quite a large number of steps that we had to go through and tease out exactly how each of these were coupled together, what the dependencies were, and then put it in a modern workflow language, such as described before, the workflow description language, WDL, that allows us to run each of these concurrently on HPC, and obviously leading to that reduction in time. So it goes through a whole stage of choosing which of the sample whole genomes you've got would be your best reference genome, as well as going through the whole sequences and doing some QC, choosing the best reference genome, doing the variants, and then having the flexibility to choose specific types of variant calling and cleaning afterwards. And then once you've done that, running through and doing a final filtering, and then having a collection of variants that you can then go on and do further analysis. And particularly with this collaboration, the further analysis is understanding population structure, particularly in the agricultural environment. This has been a fantastic collaboration, and I will hand it over to, I think, either Pasi, Neil, or Robin to give some commentary. Pasi, Robin.
Thanks. Thanks very much, Noel. Can I share the screen, or?
Yep.
Am I able to? Yep.
Yeah, you can now, Robin. Yep.
Yep. Hang on a sec. So thanks very much, Noel, and thanks everyone for participating. What I was going to do is just give you a bit of background on the program that we've been doing and just the reasoning behind what we do. So we work on parasites, and we look at the genomics and genetics of parasites with a perspective on trying to control them and diagnose infection and treat them. And so a lot of parasitic diseases of animals and humans are really, really important around the world, and they are as important, although they don't spread as quickly as COVID, but they are, in terms of impact, they are often very chronic and have a major impact around the world. So we work on things that cause diseases like these. This one here, for example, is a echinococosis caused by a dog tapeworm that gets into humans and causes massive disease. This is schistosomiasis, and these are roundworms that cause problems in probably around 1 billion people globally, if not more. And so some of these diseases are recognised by the WHO as really key diseases that we're trying to eliminate over the next 20 or 30 years. And so a lot of the work that we've done is really trying to focus on understanding these parasites at the molecular level, understanding the way they interact with the host animal and humans, obviously, using the latest technologies, and trying to identify drug and vaccine or diagnostic targets. And so this is our research program, which includes a lot of areas in the omics field. It covers, areas such as transcriptomics, proteomics, lipidomics, and so on. But we're trying to understand parasites and their populations. And when we started talking to Noel and Bobby and others, and particularly Professor Turpin, we were focused on population genetics, trying to find a better way of undertaking population genetic investigations. And we'd been doing a lot of work on genes, and we expanded to, whole genomic data sets using single copy orthologs to try and get insights into the population genetic structure. And at the time we were doing a project on the carcinogen. This is a carcinogenic parasite that induces cholangiocarcinoma in humans. It's particularly important in parts of Asia. And there's a related parasite that causes the same sort of disease that's highly endemic in other parts of Asia. So we were very keen to understand I suppose, the genome and understand how much genetic variation exists in populations of this particular parasite. And that's where we started working up a manual pipeline, which we used for this particular study. But then eventually we went on to develop this automated pipeline that Noel and Bobby have and others have helped us develop very, very well over the last period, particularly over the last 6 months. So, you know, members from our team, including Pasi Kohonen, Neil Young and Lena Kinkar, have been sort of critical in assisting with the development of this initiative. And I suppose the main point to say is that alone we couldn't have done it. Together, the project was synergistic. It was synergistic because we have, on one hand, specialists within MDAP that have enabled the construction of this pipeline under guidance from us who have had a lot of experience analysing data sets and really trying to make biological sense of the data sets. So it's been a fabulous interaction and has really facilitated and exacerbated our capacity to handle really large data sets for parasites. And I expect that we can see expansions of this particular pipeline to a lot of other areas where people are trying to look at genetic variation in parasite populations, population structure, and phylogenomics. So it's been really a great experience for us. So thanks very much to everyone at MDAP, and thanks to everyone in my team who have worked so hard to make this work so well.
That's fantastic. Thank you so much, Robin. I think, so the next slide we have, if you just want to share.
I think we'll just, I'll just mention it, Noel, that that people can go to the to our website for more information, and or email us at mdap-info@unimelb.edu.au if you have any specific questions about projects, ELIs that you're interested in. There's now 10 minutes for us to do a bit of Q&A. If people do have any burning questions and you'd like to ask whilst we're all together, please feel free to either unmute and ask the question, or you can put something in the chat for us to address.
Can I ask? Yeah, so thanks everyone. That was a really great overview of what MDAP is doing.